Greetings students, so I'm Mr. Sanger here, and what I want to do today is I want to do a quick recap of how to reconstruct that phylogenetic tree from the Ebola DNA sequences. So, let's take a look at our sequences. Okay, so the first part of this task was you were supposed to look at the DNA sequences and just kind of organize them and group them together. And so, if you did this, you may have noticed that there's 4 sequences that have 2 differences from our reference. This T that has changed to a C and this C that is mutated into a T. So all 4 of these share that. And then if you scroll down even further, you'll notice that there's 3 more sequences that still have that original, those 2 original changes, but then also have 4 more changes. The original C has mutated into a T, this A to a C, this G to an A, this T to a C. And then if we scroll down even further, we see that there's 3 more sequences that have even more mutations in here. Whoops, actually just 2 more, I guess. That's the wrong one. That should be up here. Anyway, so we see that they've inherited those same 4, but then there's this extra C mutation on the end. So you can kind of see there's been sort of 3 major mutations or phases of mutation that have kind of taken place and then spread throughout the population. This original C and T mutation, this next group of 4 that came around, and then this last group of 1. Now, in addition to that, so 15 goes up there with that, there's also some oddball sequences. I'm just going to kind of clear those to the side. But then there's just some oddball ones that didn't quite seem to fit anywhere, and they have a lot of those same mutations that we talked about, but then there's other mutations that didn't seem to spread. So like this G, this is the only one that has that mutation, and this is the only one that has that mutation. And so these oddballs we'll kind of come back to later. Those essentially are mutations that they didn't take. So those they didn't spread and become predominant. So the core ones, those first ones, were probably mutations that helped the virus infect a cell more effectively or spread more effectively or something, and then these were maybe more dead-end sort of mutations that just kind of came along and didn't help that much and therefore didn't become as common as the others. So how do you go ahead and do this? So we're going to start with a phylogenetic tree, and we're going to start with a common ancestor. So the common ancestor is always at the base of the tree. And you can assume that that common ancestor had this sequence, this reference standard. That was our oldest, most original sequence. And so what happens then is that common ancestor is going to branch. One branch is going to basically just maintain that sequence. So I'm going to call that our reference sequence. So nothing really has happened, but as long as this sequence is still out and in existence, then we need to represent this up along this horizontal line that we're going to form, because this horizontal line represents all the living variations, or all the, I shouldn't say living with viruses, but all the current variants that are out there. So this one just kind of went and just didn't really change and is still out there. But then it also branched. And over here we have this branch point where 2 key mutations happened. And those 2 key mutations are these that I mentioned before. So this T changed to a C, and this C changed to a C. So basically in position 2, we had a T change to a C, and then 3, 4, 5, 6. In position 6, we had a C change to a T. And so these mutations happened here, and this individual, this common ancestor, has those mutations. And so everything that branches from this point on should have these 2 mutations. So what I'm going to do now is I'm going to make a branch. I'm going to extend this one way out, and anybody who only has those 2 mutations are going to go in this grouping. And so those are individuals, 4, 6, 11, and 13. So 4, 6, 11, and 13 have these mutations and only these mutations. So nothing has happened. Okay? But this way, there was more activity that took place. So if we go from number 4 to, let's just pick sequence 2 here, all of a sudden we have this new grouping where there's these 4 additional mutations that have taken place. And so those 4 mutations are, at position 1, the C has changed to a T. At position, what position is this? 1, 2, 3, 4, 5, 6, 7. This A has changed to a C. At position 8, 9, 10, 11, 12, a G has changed to an A. And then at position 13, a T has changed to a C. And so again, this common ancestor has inherited all 4 of those. And then there's going to be another branching. And this branch is going to be all the sequences that only contain these additional 4 mutations. And that is, I believe, sequences 2, 5, 7, and 15. But then there's one more kind of big event here. Over here, there was another mutation represented here by this sequence. And this one also had, in that final position, position 14, that T mutate into a C. So I'm going to write 14, T has changed to a C, and there were 2 sequences that had that, and those were sequences 8 and 12. And so you can see the evolutionary journey that the virus with, the variant with sequence 8, for example, took. The common ancestor branched, the mutations, that original mutation of C and T occurred. It continued on, these additional mutations took place, and it continued on, and then that final mutation took place. So the common ancestor is passing the preceding mutations on to all of their descendants. Now the last part, and I think this is where it really gets confusing, is what about those oddball ones? So sequence 10, for example. Well, sequence 10 has that weird G, and it's the only one with that weird G. And you can look at this and you see that it has inherited the same mutations as sequence 2, right? But it hasn't inherited the sequences, or the mutation of sequence 8. And so basically that weird G and that weird A, they're kind of in this branch somewhere, right? It inherited these 4, but it didn't inherit that. So sequence 10 is actually over here. So I'm just going to, in parentheses, add sequence 10. Now you could make another branch if you want for sequence 10, since that also had 2 new mutations. But sequence 10, since those 2 mutations didn't seem to spread and take off, those probably weren't very effective for helping it spread around. And you can do that for these others. 1, 3, 9, and 14 also have some kind of weird odd duck mutations. So if you look at this, clearly this has the first 2 mutations, that C and T, but it didn't inherit those 4 new ones. And so this guy must have branched off before these 4 mutations took place. So sequence 1 inherited these 2 changes, but none of those. So it must also be over here. And so sequences 1, 3, 9, and 14 all kind of fit where sequence 1 is, where it's inherited these 2 and then had a couple other random ones. And so you could put 1, 3, 9, and 14 in different branches coming off of here. But again, I'll just kind of lump them together, sort of saying they're part of that group. So what we're seeing is these what we'll call core mutations that apparently were successful enough to allow the virus to spread more effectively and give rise to kind of a new variant. And this is exactly what we're seeing with COVID, as certain mutations help it become more common, spread more effectively, and then become more prevalent in the environment. So hopefully that clears this up. Thanks.